This is Audiommunity, a podcast about our body's never-ending fight with the outside world. Hi everyone, this is Audiommunity, recorded on June 14th, 2016. I'm Kevin Bonham, and I'm joined, as always, by Matt Woodruff.
Hello again.
Who is a postdoc, at least for the near future, at Emory University.
Till he gets fired. No,
Till he gets fired.
I might get fired. That's fine.
And that other voice you hear is Kate Franz.
Hello.
Graduate student at in the Harvard Medical School virology program. And we're recording here on June 14th, and we're repeating an episode that we tried to record 2 weeks ago, but I screwed up my audio settings, and it sounded like I was recording from underneath a lake.
Beyond the grave.
And
I actually think we should celebrate Kevin messing up, because now we can just hold it over his head forever.
Yay!
Yeah, that'll be great.
I was going to do the air horn noise.
I can go find that on the internet and put it in post. But yeah, so I tried really hard to recover the audio, and I even contemplated re-recording just my section.
I would have preferred
that. But that was going to be terrible. Yeah, that would have been terrible. Wasn't going to happen. I recorded like 2 minutes of this, and I was like, you know what? This is bull. So we're going to try again. So the paper that we tried last time, and we're going to do again, is a paper that none of us really were super on top of, and I feel like it's not going to be any better this time.
I actually think it's going to be worse because
we don't have the joy of discovery this time.
Right.
True.
I think that it's there's a lot of evolution here, and I think we should probably hit all of the basic topics of evolution again, which would be good. But while I was reading this paper for the first time, I was like, oh, I should really brush up on some of my evolution terminology. And so I did that, and then had a, bad but salvageable discussion about what I thought about evolution. And there's almost no chance that I will remember any of that now.
Okay.
So that's basically what I'm throwing out there.
Cool.
Yep.
Before we get into that, though, by the way, you should be ashamed of yourself that you're a biologist and you don't know evolution. But,
hey.
He doesn't know the ticky-tacky things of evolution. He knows it's
I'm
real.
just kidding.
Yeah, my evolution knowledge usually takes the form of a germinal center. So, natural selection is constantly moving through, but purifying selection...
But that's microevolution, Matt, not macroevolution.
But fortunately,
I
Matt
forgot
is
I don't
not...
believe in macro.
I went to a high school where, unfortunately, among my peers, evolution was not an accepted fact. I hope that I'm just estimating how many people really didn't believe in evolution, but the log line was microevolution is real, but microevolution can't lead to large speciation events like macroevolution does. Macroevolution is wrong. It was very depressing.
I'm sorry.
Yeah.
That sounds horrible.
Yeah, baby
I might have
Kate
killed
did
someone.
not like high school all that much. Actually,
This is what happens when you go to high school in East San Diego.
You say that with such derision, like everyone else from
Yes.
San Diego describes my hometown, and I will just let you know, apart from our highly conservative politics and questionable views on science, it's a very lovely place to grow up and spend 10 minutes to drive through on your way to Julian, which is what everyone says. Oh yeah, I've been there. It's on the way to Julian. F Julian. They just have apples. I hate Julian. They have meth and apples. Which were you there
The
for?
only thing I remember about Julian is a giant frigging fire that happened when I was in college.
Yeah, I was in high school.
It nearly burned. That was one of the times when it was Armageddon, black sky at noon. Whoa, the lights just went off on Kate's room. Awesome.
Was this 2003 or 2007?
I don't remember. One of the two. I was in San Diego for both of those. So
I feel like we're underselling apples here. I really like apples.
Oh boy. Okay.
Okay, let's get
apples.
back on track and talk about what we're drinking this evening.
Kevin's tried to transition away from this 3 times. I'm like, nope, I'm going right back.
Coming back.
I am drinking my last Cisco Brewers Grey Lady that I purchased 3 months ago for this podcast. Magically, it was still in the refrigerator. I mentioned this last week, or the last time we tried to record, but no one's going to hear it. that I put a little sticky note on the 6-pack in the fridge asking people not to steal it, and they didn't. Furthermore, 2 Bud Lights appeared in the 6-pack as if by magic.
Nice.
Maybe people felt bad for previous beers they were pretty sure they stole from someone, and they realized that that someone sometimes left sticky notes on things.
That seems plausible, but
in which
if
case
they
they did it in the
beer that I
shittiest way
have
possible.
with Bud Light, yeah. So yeah.
If you knew the people who steal beer from that refrigerator with no regard for who it belongs to, people like me, they're garbage people who are just going to give you Bud Light and they don't give a shit.
Yeah, that's clearly the case. Also, just reminder to myself, my future self, to make sure I go back and
Right.
scrub this audio of all the cursing that just happened.
Yep.
Matt, what are you drinking this evening?
You started it.
I
Kevin.
have nothing. I have nothing.
You're pulling a Kate.
Yeah, last time I was drinking a Guinness, and this time I failed to secure a Guinness prior to starting this podcast, and so here I am.
Do you have any in the house? We can wait.
No, I don't. Sadly, I don't. I had planned on getting a Guinness, and then I just didn't do it.
Well, all right then.
Yep.
Kate, you are for once not failing us.
I know. So
What are you drinking this
I looked
evening?
into the fridge, and I was like, "Oh man, I want something really refreshing. How about that Bud Light Lime?" And then I thought Kevin and probably Matt are gonna laugh at me for drinking Bud Light Lime at 6:00 at night, first drink of the day. So then I was like, okay, I'll do something classy. I'll drink the Mayflower Porter. And then I was like, that's too classy for me right now. Like, I'm just not feeling the it's heavier. So I met somewhere in the middle, and I'm drinking a Pacifico, which is one of my favorite beers.
That's
in the middle of Bud
in
Light
the middle of Bud Light
and,
Lime
and Mayflower Porter,
and Mayflower
right?
Porter.
I mean, I guess.
Pacifico
right?
comes in a
Sure.
bottle. Like, it's a classy.
It's not a clear bottle either,
Yeah, it's
so
a classy beer.
that's good news. Yeah. I mean,
it is. I guess it is in between.
Yeah.
it's yeah. I would definitely say that Bud Light Lime is at one extreme, and Mayflower Porter is significantly further on the axis in the other direction. Like, the terrible to good ratio. I don't know where Pacifico stands. It's certainly a reasonable Mexican beer.
That's fine.
I missed all
say
of what
better
you
than
just said
Corona,
'cause my
not as
internet
good
is
as
being
Negra
weird,
Modelo.
but I just want to say I love Bud Light
I like
Lime,
Negra
and there's
Modelo.
nothing that you can say that will ever change my mind.
Anyway, beer snobbery aside,
Yeah.
today, for the second time, we are going to attempt to muddle our way through a paper that was published in eLife last month. It's called Viruses Are a Dominant Driver of Protein Adaptation in Mammals. And the authors are David Enard, Lee Kay, Karina Gwinnap, and Dmitry Petrov. And the title sort of says it all, but they do a lot of cool work to basically, and it's all bioinformatic. They're using DNA sequences that are in public databases. They don't do any bench experiments as far as I can tell. But they show pretty convincingly, I think, something that is not terribly surprising, but also useful to know, which is that viruses are driving adaptation in their hosts, at least in mammalian hosts. So if I remember correctly, Kate volunteered to do figures 1 and 2 the last time, and so I thought I'd throw her under the bus and see if she remembers this time.
Yes. Sorry, my internet is being really ridiculous right now, and I can't... Okay, here we go. Here's the paper. Well, I think I remember figure 1 off the top of my head. I believe that figure 1 is a tree of all of the mammals that were used in the actual analysis of proteins. And I remember that we had a discussion. Now this is going to be like the worst podcast ever because we're just going to be like, I remember this one time that no one else has been aware of. Yeah, we'll pretend it's the first time.
I think.
But this is... What did we say? So this is clearly... So we said that there are 2 types of trees. One of them is based on actual data and some sort of
No. Okay. So we're talking about an evolutionary tree, which you've probably seen them before, but if just in case you haven't, just in case you haven't, it's basically like a family tree that shows last common ancestor between a whole group, a big group of species. You can draw a cladogram in principle between every organism that lives on the planet, and people have done these sorts of things because we all share a common ancestor at some point, although it gets a little muddled for bacteria. But so the two different types of trees you can have are a phylogram versus a cladogram.
I remembered.
And
I hope you didn't cut out my cladogram claim.
I got that.
Good.
Good. So the difference between them is that a cladogram just shows you the relationship. So, for instance, it'll show humans and chimpanzees are off on a branch together, closely related to, say, bonobos and other great apes. And then, further down, depending on what organisms you're looking at, you might see rodents, you might see other mammals. And if you go way back, which this paper doesn't do, but you can go way back and you can, throw on fish and reptiles and birds and stuff.
So what data is a cladogram based on?
It depends. So you can make a cladogram or a phylogram for an individual protein, for an individual gene, or you can do it for a species as a whole. And the way it's usually done for a species as a whole is you'll basically take either one or a number of genes that are assumed to be relatively slow to evolve, usually really important functional proteins. Like, for example, in mammals, you might use hemoglobin or some other protein that is very important. Its structure matters.
Ribosomal
so
proteins.
Yeah, you can use ribosomal proteins. That's often used particularly in microbes. And so you can do this for individual species or for individual genes. A cladogram just shows you the relationship from one organism to another. A phylogram has branch lengths that are proportional to the relationship. So in a phylogram, humans and chimps are going to be real close together compared to humans and birds, for example. Whereas on a cladogram, if you had humans, chimps, and birds, they would all be the same distance away. Birds would be further apart, like would be on the outer branch, but the length of the branch would not tell you anything. So this figure is a cladogram, so branch lengths don't matter, which I find a little bit annoying just because there's an opportunity to present information that they just don't present.
Yeah, I don't know, though. I think that for the purposes of this paper, really all you're looking for is generalized relationships. And I think that the problem with a phylogram, when you're looking at distant species, when you have a huge variation in the amount of distance between two things that you're trying to show, is that you basically lose all of your resolution, right? If you have a couple outliers that are super far away from what you're trying to compare, and everything else is not an outlier, you basically lose all your resolution as far as the comparison between two groups.
Yeah, that's true. But I mean, it would be sort of useful to show, like, all of the primates on here are much closer together than all of the other outgroups. But
sure,
whatever. It doesn't matter. This is not an important part. It's kind of amazing that they used an entire figure for this one cladogram, which, you could have found this same cladogram on Wikipedia probably if you Googled it.
Also, does it matter? Does it really matter how close these things are related to each other? Because really, they're just using them for sets of genes.
Yes.
Right? They're looking for
further.
Yeah. Later on, when they compare humans to chimps and, to great apes, I understand that. But really, the inclusion of the microbat in this cladogram is not useful for the rest of the paper, except when you go into like figure 7 and you're talking about specific genes for specific species. But
right.
Anyway, I think...
I think the one nice thing about this cladogram and like basing it in this evolution is that so they look at different viral proteins interacting with different mammalian proteins. And so the viruses themselves also have have a relationship to the different mammals. So like different viruses only appeared or like only don't have any other and like don't have any related viruses outside of a particular group, and that has implications.
Mm-hmm.
So for like their actual point, which I'll hit on more when we actually get to it, so I don't.
Right.
Yeah.
So let's do figure 2 is also sort of a data poor figure, but the point of figures 1 and 2 is basically establishing what they are comparing. And so what they're going to be doing throughout this paper is to look at very important persons, VIPs,
or proteins. Very important proteins.
Virus interacting proteins is what this refers to. And basically what they did was they curated a giant list of mammalian proteins where there is some evidence, not just some evidence, where there is experimental wet lab. They specifically point out this is low throughput experiments.
Low throughput.
That
a lot of judgment in that word.
Definitely. That indicate an interaction of this protein with some viral protein. So they excluded stuff where there's only bioinformatic evidence of interaction because, you know, there's biases that might
like
creep into that. They didn't want to deal with it. So large scale screens. If there wasn't some low throughput, which I think is just anything that's not high throughput. So normal throughput.
The first time we recorded this, we spent about 20 minutes debating what that term meant, and I don't think we should do that again. But I'd like to reiterate the point that just because something's high throughput, it doesn't make everything previously done low throughput. It makes it normal throughput, right? We don't have to place it on the other extreme just because something faster has come up.
Low throughput is everything that was done before the time that whoever you are were in graduate school.
Right.
So
I'm sorry, but I can only think in binary terms. So
I see.
I'm going to have a problem with
So
this.
we're going to call it, this year will be 3BS, which is before or after screen.
Yep.
Or a bunch of bold.
Yes.
See, and then,
Right.
Yeah.
I saved you one,
Thanks.
Kevin.
I appreciate that. No, I was going to say, like, it feels to me like anything that was done before, like, around 2006, 2008 was low throughput, and everything afterwards is high throughput. And I imagine someone that's in graduate school now is like, microRNA? That's so old school. Like, you know, we've got RNA-seq and single cell, all this crap. But if you were a scientist and you went to grad school in, like, the 60s or something, like, a PCR is going to seem super high throughput compared to, like,
right. Or DNA
dipping
sequencing.
your tubes in water baths. Anyway, so figure 2 is basically just a chart of the number of virus interacting proteins discovered over time from 1995 to 2014. Again, not super data rich figure. I really feel like these first two could have gone in the supplement, but whatever. They're sort of establishing what they're doing. So they're looking at these virus interacting proteins that they've pulled from public databases that have evidence from some wet bench experiment that they actually have some interaction with a virus.
Right. And they do mention, I think, that they stopped pulling these virus interacting proteins in 2014, and basically say, you know, if you look at the number of proteins that have been identified, you know, by the time we stopped looking through the literature, they were being discovered at an increasing rate. So the likelihood that more of these proteins will be found only goes up as time progresses, which means that they're probably underestimating the number of viral interacting proteins, which means probably their statistics will only get better as time goes on.
Right. I imagine that they started this project in around mid-2014, and at some point you just have to make a cutoff and say, okay, this is going to be my data set.
Sure.
And it probably took them a little while to actually churn through the analysis. Okay, so the other thing, just by way of introduction, that I want to do before we get into the real meat of the paper is to talk about this measure of gene similarity. And this is the PN to PS ratio. And what that stands for is when we're looking at a nucleotide sequence for a protein coding gene, some of the mutations that occur in that protein are going to, or rather, some of the mutations that occur in the gene are not going to affect the sequence of the protein that is coded for by that gene. So we talk or biologists often talk about the redundant protein code. And what this means is that each amino acid in a protein is coded for by something called a codon, which is 3 DNA nucleotides. So in a protein coding gene, every 3 DNA nucleotides is the code for a single amino acid. But there are only 20 amino acids that the DNA needs to code for, and you can do the math. 4 bases, 3 bases.
3 bases in length.
3 nucleotides in a codon, each of which can be one of 4 bases, gives you 64 possible 3-letter words, essentially. So we have 20 amino acids that we need to code for plus stop codon. So essentially 21 things we need to code for. And we have 64 possibilities. So what ends up happening is that a lot of codons have, or a lot of amino acids have multiple codons, multiple 3-letter sequences that code for them. And the way it works out is that often it's the 3rd base of the codon that can be variable. So there's a lot of amino acids where, for example, I should probably pull one up so I don't just make one off the top of my head. But you can Google codon table and find some of these pairs. But for example, I just pulled something off Wikipedia. The code CTT, CTC, CTA, and CTG all code for leucine. And actually, it looks like TTA and TTG also code for leucine. So there's a lot of redundancy like this. Some amino acids have more redundancy than others. But what this means is that some DNA mutations, even though we can see them in the evolutionary record or in a DNA sequencing record, don't actually affect the protein. So that mutation is essentially silent. Silent mutations, for the most part, are not going to be acted on by selection. So we can use the ratio of silent mutations or synonymous mutations compared to the ratio of non-synonymous mutations. And that can tell us something about the evolutionary history of this protein, whether it's under positive selection or negative selection.
Right. So, yeah, because the underlying assumption, I guess, here then is that codon mutation is going to happen at a relatively stochastic rate, and it's going to be totally agnostic to whether the mutation is synonymous or non-synonymous. So the real question is, when you get a non-synonymous mutation, does that non-synonymous mutation have a large impact on the organism's success, right? And so if you see a lot of synonymous mutations, which means that the protein has been hit a lot of times by mutation, but you don't see any non-synonymous mutation, it tells you that if you get a non-synonymous mutation, you're changing the functionality of that protein, which is not viable in that organism.
Right. So another way to think about that, if you just count the synonymous mutations, if you just count the mutations that don't affect proteins, and you compare, say, 2 organisms, let's say you compare me and Matt versus me and a chimpanzee in Africa, and you look at just the synonymous mutations, just the mutations that do not affect the protein function. What you can do is you'll see that there are going to be more synonymous mutations between me and the chimp compared to me and Matt, right? That suggests that Matt and I are more closely related to each other than me and a chimp.
Yeah, although I don't know that the data would work out that way. I just called you a chimp.
Oh, I see. Well done,
get
Matt.
It? There you go.
Okay, so
the slow pickup.
Yeah, I just
right.
was like,
It
wait a
was a
minute.
deep
Are you
science
sure
joke.
that data? I think the data would work out that way. Anyway, so just looking at the synonymous mutations, which ends up being the denominator of this ratio, the PN to PS ratio, that gives you some sense of the time, the evolutionary relationship between 2 species. So for an individual protein, if you see a high ratio where there's a lot of non-synonymous mutations relative to the number of synonymous mutations, that gives you a sense that this protein is not under strong selection, right? Because this protein can accommodate lots and lots of different mutations that are altering the function of the protein. It doesn't much matter. So we're going to see in an intron, for example, that doesn't code for a protein, but let's just say in a protein where there's a duplicate, so you have redundancy, one of those proteins can accommodate a ton of non-synonymous mutations. It can just accumulate mutations willy-nilly because it's got another protein that can back it up. But for a really important protein like hemoglobin, which I mentioned before, most non-synonymous mutations are going to disrupt the function of that protein, and that protein is super vital for life. So disruption of function is going to have really deleterious effects and are going to be selected against by natural selection. Yes? Do we all agree with this?
Yep, I buy that. I like the word
Yes.
accommodate for this. If the protein can accommodate mutation without harming the organism, it is not going to be it's not going to be under a lot of selective pressure.
Right.
Yep.
So in figure 3, what they're doing is they're basically looking at this PN to PS ratio for the virus interacting proteins compared to a huge panel of proteins that have no evidence of virus interaction. And what they find is that the proteins that are interacting with viruses tend to have a very high PN to PS ratio, which means that there are a lot of mutations relative to, or
It's low, right? Low PN to PS ratios?
Yeah.
Yeah. I'm certain it is.
I'm mixing up the blue and the red. Yes. That is correct. God damn it. This is what happened last time. Okay. There's no legend on the panel that I'm looking at, so that screwed me up.
Yeah.
So there's a very, very low PN to PS ratio for the virus interacting proteins, which suggests that most of the proteins that are interacting with viruses are under strong selective pressure. Yes?
Yes.
We agree with this. Okay. And one of the explanations for this is that viruses tend to interact with proteins that have a hard time mutating, because if the protein had an easy time mutating, then populations would rapidly accumulate mutations that would prevent virus interaction with them, right? Because most of the time when a virus interacts with a protein, it's either because it needs that protein for part of its life cycle, so getting rid of the virus interaction would disrupt the viral life cycle, or viruses are interacting with proteins in order to screw up the host response to that virus. So if you can avoid, for example, in an immune gene, in an innate immune receptor or something, if a virus could interact with that to disrupt it, then evolving away from that binding would help
So
evade the virus.
I agree with the general concept that you're saying, but I don't think that this is what the data really... I don't think that's the only model that this data bears out, because I think implicit in that is the assumption that the species has the ability to mutate away from the viral stress. Whereas the viral pressure, unless you're killing the infected host at like a pretty phenomenal rate, it's hard to these viruses are somewhat new. Like I would say it's hard for the host to adapt to the virus if there isn't like a very strong pressure for the host to do so. Whereas I think what you could say is that the virus is utilizing proteins that are essential to the host. So like the host can't. Okay, sorry. It's like getting to the same point. Like, but the virus interacts with essential host genes, and they're not going to take any mutation. Not that the virus really could change them, like make them mutate. Like these are genes that can be conserved, like almost from bacteria.
The way that I'm thinking about it is sort of as the reverse of what we talk about with innate immune receptors. So we often talk about the idea that innate immune receptors that Kate and I both studied in grad school tend to target components of pathogens that are difficult for those pathogens to get rid of that are essential to the pathogen
Yeah.
survival.
But
So
also
like
because-
LPS or like genomic content. And so I'm thinking sort of the reverse of that in terms of viral interacting proteins. One interpretation of this idea that most of the viral interacting proteins have a low PN to PS ratio is that the proteins that viruses are evolving to interact with can't mutate as much as proteins in general. And one of the reasons for that might be because viruses that are interacting with proteins that are easy to mutate away from are not going to survive very well because any selective pressure they put on the host will mutate away. I don't think that's what you're talking about. What you're talking about is the fact that viruses, part of their life cycle is taking over essential machinery of the host, like stuff involved in transcription, translation, and just the nature of that relationship, what they need to parasitize, is what is causing this different ratio.
But how would that? Sorry. So how would that selection work then? So you have, I think that model, and maybe I'm just thinking about this wrong, but I think that model would suggest that if you mutated away from. So first of all, it's worth talking about figure 3C, right? So figure 3C basically breaks all of those viral interacting proteins down into different kinds of proteins. So it's like classes of proteins.
Right.
And basically what that figure shows is that there is conservation across a huge set of genes. So across a really broad set of functions in the host, as long as those proteins are interacting with the virus, you do see this viral pressure. So presumably there are a lot of really critical proteins in those pathways that are not interacting with viruses, and those are not being touched by this interaction. Just because the virus is interacting with that pathway doesn't necessarily mean that the protein is going to be conserved. Right? So wouldn't that suggest then that the only real explanation is that these proteins, because they interact with viruses, if they mutate, it causes some sort of lethality in the host on that viral interaction?
I'm not sure the causality, like how you're drawing the causality arrow. So the way that I'm interpreting this is, in a typical host genome, there are going to be proteins that can accommodate mutations, and there are going to be proteins that can't accommodate mutations as well.
Sure.
Right?
So let's assume two critical proteins in cell division. All right, protein 1, protein 2. Protein 1 and protein 2 are required for cell division function, right?
Mm-hmm.
A strong mutation, a non-functional mutation in either, is going to give you a problem in cell division. Was I saying cell division before?
Mm-hmm. Yeah.
So a strong mutation in either is going to shut down your ability for cell one, or for the whole thing, for the entire cell to become non-functional, right? But only one is a viral interacting protein, because and what we're seeing here in figure 3 is that just the viral interacting ones are the ones that are conserved.
Yeah, but this data doesn't tell us what you're saying. So let's say there are 100 proteins involved in cell division.
Yeah.
20 of them are essential and are not able to accommodate non-synonymous mutation. What this figure might show is that the 80 that are capable of accommodating mutation, viruses don't tend to interact with those proteins.
I see.
Yeah.
But the 20 that are super essential and cannot accommodate mutation, the viruses interact with that.
I see. So yeah, it's the evolutionary relationship between the virus and the host that's causing this interaction to be critical, right? So the virus is basically targeting critical points in the pathway.
Yeah. So the question is,
okay,
so
to prevent
we know that viruses tend to interact with proteins that cannot accommodate a lot of mutations. That's all this data shows.
Right.
Right. The proteins that viruses tend to interact with are less able to accommodate mutations than proteins that viruses don't interact with. It does not tell you that viruses are causing these proteins to not be able to be mutated. It doesn't tell you that. It doesn't tell you why viruses are why viruses have evolved to interact with these sort of proteins. It could be that viruses are doing this because there is a selective pressure for them to target proteins that can't change, or it could be the case that viruses just need certain types of proteins, and those proteins that they need happen to be ones that can't accommodate a lot of mutation.
Yeah, that's basically what I was trying to say.
Okay.
Okay, good. It only took us 30 minutes to get there. Excellent.
Right. So then, but then if we go back to the title, right, which is viruses are a dominant driver of protein adaptation. That sounds like a positive process that they're spinning on it. So do you disagree with that interpretation?
No. So that evidence comes later.
Okay. Yeah. So we're just not there yet. You're saying that this evidence does not point that direction, but we will get there.
Right. Exactly. So maybe we should skip ahead. So the figure 4 is showing just human viruses, and looking at viruses that tend to interact with humans. We can sort of skip over that. I think that the figure that gives evidence for the claim in the title comes in figure 5. And the idea here is we're going to look at a different way of looking, or wait, is it figure 5?
Yeah, I think it is.
I think it is figure 5.
The busted tests.
No, actually it is figure 4. No, it's figure 4. I'm sorry. So here we're looking at this value called alpha. We looked this up last time. This is a classic MK test, which you can look up on Wikipedia and try to wrap your head around what it is. But basically the way to think about it is we are comparing the PN to PS ratio within a species to the PN to PS ratio between species. So going back to what we were talking about before, if we're comparing the PN to PS ratio between me and Matt compared to between me and Microbat.
Yes.
Exactly. So the idea here is if a protein, so for those really essential proteins, so let's just keep using hemoglobin as an example. Hemoglobin over evolutionary time is not going to be able to accommodate many mutations, right? It's the structure of hemoglobin is super important. We only have one hemoglobin gene that's expressed in adulthood. At least most humans only have one. If you screw that up, you get really bad problems, and it's going to have strong selective pressure against it. Okay.
I feel a little uncomfortable using hemoglobin as our example over and over again because of the sickle cell trait, which is exactly a mutation in hemoglobin to avoid malarial infection.
Well, it's not to avoid, right? That happens to be...
It's to protect you from...
Let's do actin.
It's just not a... Yeah, thank you.
Let's do actin. Okay, so actin is a cytoskeletal protein. Super important for cell structure. Almost everything deals with actin. There are bacteria that screw with actin, so it's maybe also not a great example. But okay, actin, very highly conserved. Strong selective pressure to keep it from changing. So its PN to PS ratio is going to be very, very low. Right?
Yes.
If we compare between humans, we look at the PN to PS ratio between humans. Most humans are going to have the exact same allele of actin. Right? Mm-hmm. So if you compare our most recent common ancestor, their actin gene compared to most present-day humans, actin is going to be almost unchanged in terms of the non-synonymous mutations. There may be different alleles that have accumulated in positions that don't change the function, but there are going to be very, very few non-synonymous mutations. Now, if we compared humans, so if we compare the PN to PS ratio between species and between humans and other primates, we would also expect low PN to PS ratio. And you can sort of, for certain genes, you can build a sort of expected amount of non-synonymous mutations that are going to accumulate over time. You can sort of build a molecular clock. If we now look at a distantly related species and we see more non-synonymous mutations than you would have expected, given the slow rate of evolution, then you might say that gene is under strong selective pressure to change. Even though it is hard for that protein to accommodate changes, you would expect a higher ratio of non-synonymous mutations if it is under selection to change, right? So if there's some protein that a really terrible virus interacts with, and there's a mutation that could block the viral interaction, that mutation, even if it screws up the protein function a little bit, it might be still strongly selected for because of virus interaction. Does that make sense? I think I explained that better than I did last time, but still not very good. So
no, I think it worked. Let me massage it a little bit. So evolution happens sort of as at a fixed rate, right? The mutation rate of a certain protein can be predicted, I suppose, right? So based on the relationship between species, we should also be able to predict with some accuracy, especially in conserved genes, roughly how many protein mutations we would expect in a species that is related at distance X, right? And if we see that protein is highly conserved, but for some reason in species 2 has been heavily changed, even though we know that protein is under strong selection not to change, it suggests to us that organism has probably interacted with something where it was under dire circumstances where that protein really needed to change for the survival of that species.
Right.
Yeah?
Yes.
All right.
So basically, before we were saying that a low PN to PS ratio meant there was strong selective pressure for the protein to not change. So in figure 4, as opposed to a high alpha value means that the protein has changed more than you would have expected based on the phylogeny of that protein. Okay, which suggests that something is driving this protein to change more than it would if there was no other selective pressure on it, right? So you can sort of think of it this way. There's one selective pressure, which is just the fact that this is a important functional protein. That selective pressure is trying to prevent change. And what the alpha is telling you is it's giving you a measure of a selective pressure that is counter to that is trying to make the protein change, or rather is selecting for changes to the protein above the value that you would expect.
Right, and we're specifically seeing that pressure in proteins that have been identified to interact with viruses versus proteins that have not been interacting with viruses.
Right, so in figure 4, they're showing you, in this case, the blue line is above the red line. The blue line is for the virus interacting proteins, and so those seem to be under strong selective pressure to change, despite the fact that they're important proteins that normally resist change.
I always like audio descriptions of data because the dot is higher and to the right.
The good news is this is eLife, so this is an open access paper. You can go take a look at the data if you want to.
And you should.
And you should.
And you can also look at the Wikipedia page for Alpha, which I did 2 weeks ago.
McDonald-Crichton is the classic McDonald-Crichton test.
Right.
And the other thing that's really nice about eLife, that's not necessarily germane to this discussion, but they also most of the time publish the original versions and the reviewer comments. So you can go and see the revision history. So, which is really nice.
I think that's awful. They should never publish.
Hi, this is Kevin, recording on June 16th with a little interruption. What followed here in the original recording was a 20-minute discussion where Kate and I basically yelled at each other about open peer review and some other stuff. So I decided to cut that out. I'm going to put it in at the end of the episode after the music. So if you want to listen to it, you can, but Kate and I are also going to record a mini-sode together where we have a more hopefully productive discussion. But for now, I'm just going to jump past that whole thing and get back to the paper.
Kevin, that might be actually the only time in your life that you're compared to Ayn Rand.
I hope to God that is true.
Oh, boy.
So anyway, figure 6.
I am for a free market of ideas.
Okay,
let's move on.
Figure 6.
Okay, Ayn Rand, let's keep moving.
Moving along. That feels like most of the paper. No. Okay, so
it does feel like most of the paper.
I'm gonna jump.
Mostly
So figure 3, they show a couple of other cladograms where they show individual proteins that they pulled out of this analysis as being viral interacting proteins that have this strong apparent push from viruses to change, and they show how individual branches of the cladogram they showed in figure 1 are affected by these changes. And they seem to correlate with viruses that are particular to that clade. So they show...
Do you mean figure 6?
Yeah, that's what I'm talking about.
Okay, you said figure 3.
I was referring back to figure 1. Did I say figure 3? Whatever.
Sure did.
Okay. I was talking about figure 6.
All right.
So the thing that I think is most interesting about this paper, so they have all this bioinformatic analysis. They show, okay, there's in bulk, it seems like proteins that are interacting with viruses tend to have strong selective pressure to change more than you would expect. But then they say, okay, let's take an example of a particular protein that they pulled out of this analysis. So they pull out this protein. For some reason, it is not showing up at a good size, so I can't read it. Okay, so they pull out this protein.
Or is it that one? The coronavirus receptor?
Yeah. So they're looking at where is the name of the protein? Why can't I find it?
It's aminopeptidase N.
Yes,
N.
thank you.
Yep.
So aminopeptidase N was pulled out of this analysis, and previous studies had shown that this interacted with coronaviruses. And the nice thing about this protein, they sort of curated a bunch of their hits, and they pulled out this protein because not only do we have examples of this protein in a bunch of different species, there's evidence that this interacts with coronaviruses in multiple species, but we also have some structural information associated with this protein. And so what they could do is they could map the non-synonymous mutations that are occurring between species and compare where those non-synonymous mutations are relative to the site where this protein interacts with coronaviruses. And what they show, and there's a really pretty picture you should look at, is that the farther away you get from the place where this protein interacts with coronaviruses, the fewer non-synonymous mutations you have. So in other words, the bulk of the non-synonymous mutations, the mutations that are actually changing the amino acid, the bulk of those mutations are occurring right at or very near the site where this protein is interacting with the coronavirus, which suggests that the mutations that are the only ones that are being allowed, or the ones that are being selected for, are the ones that maybe disrupt interaction with coronaviruses. So I think this is a really strong, this is sort of an anecdote pulled from their giant, more systemic look, but this anecdote is really suggestive that their hypothesis is correct, that these proteins, even though they're under strong selective pressure to not change because of their functional properties, the fact that viruses are interacting with them is driving them to mutate away, in principle, from interacting with viruses. So this is just one example of that, but I think it's pretty cool and pretty convincing evidence that their hypothesis is at least pretty damn plausible.
Yeah. Yeah, at the end of every systems and high level, broad look paper, right? You need to actually dig into the data, and you need to show that, on a more granular level, what you are sort of inferring about this large data set turns out to be actually the case. And I think that this is a pretty, certainly, and probably pretty compelling case that it certainly might be. You'd obviously want to check a couple more proteins and see that this holds on the off chance that this is, but I like the type of analysis. I think that it's a cool approach to, confirming what the data is suggesting.
This one's easier because you have one protein that interacts with basically just one viral protein.
Right.
And that evolves over time. So it's like necessary for entry, which makes it like it should make it like pretty primed for evolution. Or
Yep.
like actually like
there's going to be a
maybe a
lot of
little bit-
selective pressure on it.
yeah, there's a lot of pressure on it because a really great way to avoid being infected by a virus is just to make sure that the virus can't get in. So like
Right.
this is a very selected step in viral life cycle.
Yeah.
So whereas like the other proteins, other VIPs are going to interact with multiple viral proteins. So you might be seeing a mixture of pressures from different virus proteins. Exactly how they interact with that protein is going to be slightly different.
Right. And we should say that it's entirely possible that a bunch of the virus interacting proteins they pulled out may not have any measurable effect on evolution,
That's
but
probably true.
most of their information is in the aggregate, where you're looking in general. Proteins that have evidence of interacting with viruses tend to have strong selective pressure on them. And we should also say, as Matt mentioned up at the top, that there's probably a lot of proteins that we don't know are interacting with viruses that were lumped into the virus interacting proteins in this study because no one's shown an interaction before.
Yeah.
So as we accumulate more and more, maybe this evidence will get better and better.
Because even though most viruses have far fewer proteins than a mammal does, their proteins are very, very multifunctional. So
Yeah.
one protein can interact with so many cellular proteins.
Right.
It's really impressive. It's a very cool little piece of engineering.
Kate shows her
Yes.
stripes as a virologist.
Yes. I feel like
Aren't those viruses pretty?
the first time we talked about this, I think I talked more about viruses, but
That's true. I think that's true. Yeah, I apologize if we, the rant that no one's going to hear until I release the mini-sode.
you should release the Minnesota first and be like, get pumped. I was going to say,
No, it's been too long since we posted a real
get
episode.
hype.
So
A new episode is coming. I learned hype on Reddit talking about Game of Thrones, so I'm pretty excited about that.
Nice.
That was the first time you'd heard about hype?
Yeah, I didn't know what hype was.
Really?
I thought it was like hyphy.
No.
Is it
not
like
hyphy.
hyphy?
No.
Okay, I got really excited because I think whoever made the programs that they use to analyze their data, I think they're from the Bay Area.
Oh.
Their programs are called like hyphy and something else. That sounded very Bay Area to me.
Why does hyphy sound Bay Area?
Like hyphy movement? Yay area? Get dumb? No?
Sorry,
All of these are lost on me.
I'm thinking of fungal hyphy.
Yeah, that's what I was going with too.
Like sideshows?
Nope.
No, got nothing.
Sorry. Anyway, we have email. We should read the email. If you like the show, or if you have questions or comments, you should definitely let us know. There's a form on our website, or you can email comments@immunity.org. So we got an email from Anthony, who writes something in reference to Audiommunity 20, which I don't remember what we talked about on episode 20.
So I think this was also an evolution issue.
It's possible.
So one thing I want to say is that we are
Well,
aware
let's
that we
read-
did not get everything right during this episode, so save your emails.
No,
They're
don't
very
tell us when
nice.
we didn't get it right. We like getting emails, Kate. Stop it. It was about the episode with Jesse.
Yeah, Jesse. Yep.
So what he said was, I'm far from an expert. This is Anthony. I'm far from an expert on the subject, but my understanding is that the myxoma virus evolved into a less virulent form. My impression is that this was a factor in the resurgence of the Australian rabbit population. So this is referring to the virus there was an attempt to use this virus as population control for rabbits in Australia. I think we talked about the rabbit-proof fence, and then also using viruses. And so he's saying that myxoma virus evolved to a less virulent form. Did we talk about something other than that?
I think we might have said that the rabbits evolved to be resistant to myxoma. But who knows?
Probably both of these things happen to
I
some
would say
extent.
that there is some really good theories about viral evolution that would support Anthony's impression or understanding of it. So there's this theory of moderate pathogenesis that essentially says that a virus really doesn't gain anything if it's constantly killing its host. It's a very bad transmission strategy if very quickly after becoming infected with the virus, you die. So how is the virus going to propagate itself if it kills something really quickly? So like virologists call this like the virus is really hot. So like it's very pathogenic very quickly. So there's this idea that a virus is trying to evolve towards being able to be tolerated by the host and then transmitted, and that this is a successful virus if you can infect a host efficiently, and then that host doesn't feel too sick, so they walk around and they infect everyone else, just like how you get a cold. And it makes you like a little bit uncomfortable, but you survive and the virus survives to like infect you again, maybe somewhere down the line.
Herpes virus is my favorite example of this.
Yeah,
but
I think it actually works outside of viruses too, right? Isn't this true of the malaria
this is true
subtypes?
of pathogens in general. This is a common sort of arms race idea. I actually I taught a course on infectious disease at Emerson College, and this was one of the assignments that I gave my students about myxoma. And I said, the rabbit population is rebounding after crashing. How would you tell if it's because the virus has evolved to become less virulent, or if the rabbits have evolved to be more resistant, or if it's some combination of the two? And had the students design experiments to test this. And so it is.
How'd they do?
They actually did really well. And these are non-science students. These are all like arts majors.
That's awesome.
And they did really great. Yeah, I was pretty impressed by it. Anyway,
Good.
So thank you for writing in, Anthony, and keep the comments coming. He also sent us a link to a song by, is it Radio? No, not Radiohead. I opened this earlier. It is a song by Radiohead called Myxomatosis. I had not heard of it, but it sounds pretty great. It's been Audiommunity, recorded on June 14th, 2016. I'm Kevin Bonham with Matt Woodruff and Kate Franz, and you can find the show and like the show on Facebook. We're on iTunes and Google Play Music and Stitcher. And there's an RSS feed on our website if you use some other podcasting app. Although if you're listening to this, you probably already know how to find us. So I don't know why we always need to say that at the end of a podcast, but whatever. If you like the show, you should like us on Facebook, but also leave us a review in iTunes or on
5
Stitcher.
stars.
Ideally 5 stars, but
Not ideally.
I think we
bother
begged
with the review.
on the first. I think we begged on the first take, and I think actually Kate tried to coerce our listeners into 5-star
I think
reviews.
I said, don't even bother unless it's a 5-star review. Why do you go around like spreading your haterade? No. Thank you for listening this far. If you have listened this far, I assume you like us.
And also, we are now on Microbe TV. Vincent Racaniello's
Microbe TV.
website.
Sorry
so
for all the
we
cursing.
are featured. Don't worry, I'll bleep it all out. So thank you to Vincent for letting us in and trying to capture some of that sweet Twiv audience. That sounded really gross, but I'm gonna leave it in anyway.
It does. We're just gonna keep moving forward.
And real grovelly.
Moving right along.
Also, before we go, I want to plug another Harvard podcast. It's done by Harvard's Science in the News. It's a student-run organization, and they promote science topics and science understanding among people around in the community. And they have a new podcast called Sit and Listen, which is really awesome. And
almost as funny as our name.
Has a hilarious name. And
we should make sure this is clear. It's SITN because science in the news.
Oh yeah.
The acronym is SITN, so it's SITN listen.
Yes.
Right. Visual puns work well on podcasts.
Yes.
That's what I've learned.
But they have some really cool episodes. I was supposed to go to an episode taping and I got really sick. So I was sad, but they had one where they interviewed the millennial outreach coordinator of Monsanto, which I would recommend.
Which was a fascinating episode, by the way. You should definitely listen. We'll post a link in the show notes to that as well as.
But support other Harvard podcasts.
Yeah.
So I think we should form a Harvard podcast network. Which is only like slightly pretentious, but I mean, like, whatever.
No, it's the most pretentious.
We just joined Microbe TV podcast network, so we're not going to now make a splinter group.
Yeah.
Sorry.
Okay. Fine. There's also one about
cage streams.
They published a new episode recently, and it's about mosquitoes. And there are virology students who also will talk to you about mosquitoes and mosquito-borne illnesses. So there's some more virology love out there too.
The music at the beginning and the end of the show was composed by Rachel Reineck, and we'll see you next time.
Yep.
Which is really nice.
I think that's awful. They should never publish the reviewers' comments.
Really?
They're unpeer-reviewed. People will now start to cite reviewers' comments or read reviewers' comments and think that they are correct, but reviewers' comments are not peer-reviewed. So
neither are preprints. Neither are conference posters.
You can't publish conference posters. Like, you have to wait for the paper. You don't cite conference posters and papers.
Here's my counter argument to that.
There is plenty of science that is on the internet that is not peer reviewed. But you can tell the difference between peer reviewed and non-peer reviewed things.
Yes, but comments, I don't think that people are going to. They're going to cite papers and say, I know XYZ fact from the paper, but they actually are reading a reviewer comment. I also think it makes people lazy. Because now they're like, oh, what did the reviewers have a problem with this paper? Oh, I'm going to say that's a problem too. Whereas the reviewer might be wrong. Like, that's the thing. It's just like the reviewer comments are they're not likely, they're not
I'm sorry,
they're not
I need
like
to,
going to be true. They don't have to be true.
This is a tangent, but I feel strongly about this. So peer-reviewed papers can also be wrong.
Yes, of course they can be wrong. But
so,
they could be wrong in interpretation of the data. They could be wrong because they're like just false and someone lied, which you can't really do anything about. That's going to pass peer review. They could be wrong.
They can have shoddy statistics. They can have, there's plenty of things that, so we should be skeptical of everything. There's no reason to not put information. But that's,
I don't think it's fair to put peer review comments on papers. Why would you put that on there?
So you can see what experts thought about the paper.
Why do you need to know what someone may or may not thought about the paper?
Because then you can go in and see, like, if you're not an expert in the field, you can see what experts in the field thought about it. If
you don't know who they are,
they publish the names of the people.
Oh, do they put even the names here?
Yeah, they're open peer review.
So that is the part that I actually like. So the thing is that I think that there's a big problem where reviewers are not held accountable for crazy bashing of papers unnecessarily for their own personal gain.
Yeah.
And the one problem that I think that this solves is if you have one reviewer that's constantly posting just like awful things about an entire field, this could definitely curb that a little bit because at least their name is associated with that bashing. I agree with you that it makes me a little bit uneasy for a first-year grad student to look at someone else's comments and say, oh, well, they didn't believe this because of X, right? It does make it a little bit easy, but I do
grad students are the laziest people. I know that, like, they'll be mad at me for saying that, but, like, they're so lazy in their thinking.
But, okay, so I feel really strongly about this. I'm surprised that you guys are against this. So
I think that, like, what Matt's talking about, what it should be is that all the reviewers know who they are, and that they read the papers without the author's names on it. I think that solves a lot of problems.
I think that would help. So there's a few problems with peer review. So one of them is that people don't get credit for peer review, right? So, like, if I've peer reviewed 20 papers, nobody knows that I've peer reviewed 20 papers, and it's kind of a terrible thing that journals get this work from scientists basically for free, and there's no credit given to it. So I think that's a bad thing. Another bad thing about it is what Matt was talking about with anonymous peer review. People can just all over a paper without having any consequences. But also, if you are a grad student and you are learning how to read papers, you're only reading published peer-reviewed papers, and you're expected to critique that somehow. So you're critiquing and sometimes having really, really like unreasonable expectations. You're reading a Nature paper and finding all the faults in it, which is a good thing, but it's not realistic because that's a paper that made it through peer review, right? So how do you learn what you are supposed to look for in a paper that's submitted if you can't see the crappy versions that needed improvement?
Okay, here's why I don't like peer review.
There's tons of reasons not to like peer review.
No. I don't like open peer review. Sorry, I like peer review. I brought you in with like a really hot take, and then I simmered it down a little.
Coming in hot.
Okay, so here's the thing that I believe about papers. I believe that when you write a paper, you are giving your model and you have full control. You should have full control over the data presented, how you present that data, and how you write your paper. And I think that you should be the one that dictates the narrative. That's the best thing about writing your own paper is you get to say what's in it. So I think now that when you start putting these reviewer comments on, you have people who might not be getting what the whole point of your paper is, start going on like crazy tangents, may or may not be right in the analysis that they're giving you, may be citing facts that aren't cited correctly, which then are perpetually put into the literature. So now we have like these aberrant, like these like little random facts that like people just take for truth, but we're actually like a one-off comment from a review.
Who are these people that are taking things for truth that you're worried about?
Oh my god. Okay, so
I mean,
like there's this joke. There's
Yeah.
this joke that there are a bunch of bacteria that are said to be. You know about this, Kevin. We talked about this in lab. There are a bunch of bacteria that are said to be like sensed through like particular pathways. And it's actually, and the fact has been perpetuated through reviews. And if you trace it all the way back, it's like a misinterpretation of like a throwaway line in a review.
So don't you think that it would be way better if you could go to a paper where somebody cited that in a review? The review is open.
Now we're like actually mixing
so you're
up
citing
the
that
term
paper,
reviews.
and then you can go to that review and you can say, "Hey, what the hell are you talking about? There's no evidence that this is the case. Why are you bringing this up in this review?" If
Kevin wants papers to be forums where everyone gets to talk.
No,
absolutely.
It's my story.
There's a stark contrast between what the two of you want, and I don't think we're going to come
that
to a
like
consensus
you are
here.
the person who knows your data the best, and you are in charge of interpreting it and then publishing it.
Yes.
It isn't at all. It isn't just like when you actually read the paper, it shouldn't have everyone else's thoughts on it. You should come up with your own thoughts.
I'm not saying that they can edit your paper like it's a Google document. I'm saying there is a place.
But wouldn't you like that, Kevin?
Yeah, I know, right? Kevin also believes in
so
like
long
open
as there was
lab
track
notebooks.
changes.
I do. I don't think, I mean, we're not saying that papers are going to be wiki articles where anyone can go in. Although, even if we did, like, you can see the revision history of a wiki article.
I knew you were going to still approve that.
No.
Scientists are capable of interpreting what is correct.
Crap published with my scientific study, something that they've probably thrown to a grad student or a postdoc who may or may not know what the hell is going on, and then is like written a review and told me to do 6 more experiments.
And if they do, you can say in public why they're full of shit.
Right. It is true. It is true that that one potential problem with that would be that if you hand the paper off, so let's say a big name person, let's say David Baltimore gets, pushed a paper from Nature, and they say, "Hey, review this," right? And David Baltimore, I'm sure that he reviews his own papers, but just to use him as an example, let's say that he pushes it off to a postdoc, which is a totally reasonable thing that a lot of big PIs do.
Which happens all of the time.
Which happens all the time. So then when that gets published, it's got David Baltimore's name on it as far as the comments are concerned. So people may give it undue weight largely because of the name associated when it's not actually their work. But that is another problem with the general review process.
Just so it doesn't sound like we're saying that this is a bad thing, you have to do that because you part of the training process is if you're a PI, you need to train your postdocs and grad students to review papers.
I want to be clear here. I think that what should happen in that case is there should be a separate review that is authored by David Baltimore as the last author. His postdoc is the first author. It's another piece of scholarship that is associated with that document. Yes, it is. Of course it is. Of course it is.
I agree that it would be a piece of scholarship. However, I don't think that it's realistic for any large PI to stop associating only their name with these reviews. I think that there's way too much ego associated with the fact that
if David Baltimore is consistently kicking off his reviews to postdocs that don't know what they're talking about, he's going to get a reputation for publishing reviews, and his opinion is going to, people are going to have a worse opinion of him. Part of science is public discourse. I don't think David Baltimore. He's a great scientist.
David Baltimore is. This is not an actual.
We're using him as sort of the like a paradigm of virtue at this point.
Right.
But the point is that in science we have public conversations about data. We should be having public conversations about data. We should be willing to stick to our. If I want to say that a particular piece of data is crap, I should be willing to attach my name to it. I shouldn't be able to hold a paper in review for 12 months, asking for bullshit experiments, asking for 30 mice to be made and bred, just to hold that paper back. And in the meantime, all of the data that is relevant in that paper, no one has access to it except for the authors. That's bullshit. That's bullshit.
We should get back to the paper that we are
I'm
discussing.
going to cut off all of this and publish it as a separate mini-sode.
Yeah, we should.
No, but I just want to say one thing. I want to say, like, I understand what you're saying, Kevin, but I think where we differ in what is the purpose of publishing a paper and like what should the format of that be. Whereas I think that like the paper is the one place for the author to have like the first and final say, and you think that it should be like an open discussion, and I just think you're so wrong. You could not be more wrong.
So if you publish a paper, a paper, there's a PDF of that paper.
Yeah.
Do you think that I should not be able to write a blog post, go on PubMed Commons and leave a comment on the PubMed space?
Go ahead. Read PubMed. You can be like one of the 5 people.
Do you
who
think
uses
that I
PubMed
should not
Commons.
be? No, but I mean, it's a problem that nobody uses it. But like, do you think that I shouldn't be allowed to write a blog post about it? Do you think that I shouldn't be able to talk about it at a conference?
Can write a blog post about it, but and you can talk about it at a conference. But like all of those things should not be attached to how the paper is consumed. Like when you download the PDF and how you consume this like piece of work should not be done with like a bunch of like bull crap that's been written on by like everyone and their mother, given like whether or not they like know what they're talking about.
So
or
imagine
that's
I'm
like
looking
that's for the internet. Like
right,
that's a function
but it doesn't
of the
have
internet.
to be. It doesn't have to be attached to the PDF that you download as the permanent record, but it should be associated with it.
But the open reviews
if I write a comment on a blog, it should link to the paper. And if I'm a scientist and I'm like, oh, here's a paper, I'm interested in what other people think about it, I should be able to find all of the commentary on that paper.
But also the reviews, I think that is like slightly different than having an open review process, because the review process, you're looking at different versions of a paper, and like the paper evolves with time. Mm-hmm. So like the reviewer comments don't always correspond to something that's still in the paper, or may like reference back to something else that's not currently there.
Right, and all of that should be in the record.
And I think that takes away from like the narrative that you are trying to write. Like having references to like things that aren't there, or like tangents in the actual paper.
But they're going to be there in the record. You're going to be able to look at version 1 of the paper.
But like you may take away or add data in like an earlier.
Sure.
And so like there's no reason to have that public.
Why not?
Why would you? Because you might not, you don't even know what you're talking about now, because you don't see it. Because like maybe there was like a slightly different conclusion in an original paper. Why do you need to know about that in the review? This is what I think about it.
There's going to be version 1 of the paper. There's the review comments. Then there's version 2 of the paper. You can see the diff between version 1 and version 2.
No. That's
how it works on eLife. It's great.
See, that's the thing.
I don't think we're going to come to consensus on this anytime soon.
As I said, I'm gonna cut all this out. Put it in a separate mini-sode.
We're not coming to consensus.
We're gonna put it in a separate mini-sode. Kate and I can debate about this more later, and I would love to. because I care a lot about it, but that's fine.
It's clear
And
both
we'll
of you
do a
care a lot
separate
about
one
it.
for open notebooks.
Okay, sounds good.
Which I just cackle with. Sometimes when I talk about Kevin to other people, I say, Kevin thinks that all of our data should be available to the public in real time. And then we all cackle.
All of the data for my current project is available online on GitHub
the data
right now.
is the analysis.
Yep. All of
Oh my
code,
gosh, Kevin.
all of my code, all of the like previous versions of the analysis, all of it.
Oh my goodness. You're a crazy person.
It's like Ayn Rand. Like, I have fully engulfed this rabbit hole. Have you ever seen her interviews?
Yeah, no, it's awful.
No.
It's you just like watch someone struggle with the constraints that are self-imposed. Like, I've said all of these things and now I must do all of them also, and that's proving difficult for me.
It's not difficult at all, though. It's so easy.
No, I know you
Wait,
have a much easier time with it than Ayn Rand.
In this analogy.
I think I'm Ayn Rand
Kevin
in this
is.
analogy.
Okay, thank goodness.
Kevin having self-imposed a lot of things on himself
opened up
early
like
on.
this can of worms.
Anyway,
Yeah.
Anyway, figure 6. I don't even remember where we are.
Kevin, that might be actually the only time in your life that you're compared to Ayn Rand.
I hope to God that is true.
Oh boy.
So anyway, figure 6.
I am for a free market of ideas.
Okay.
Okay, let's move on.